By day, Miss Weber is a biology teacher at Albemarle High School.
This is what we need to focus on. Okay? All right. Are you good? Thank you.
But when the bell rings, she becomes something much, much more. That's when she transforms into Dr. Science. She searches the globe for interesting research to bring back to her students. Today, Dr. Science will sit down with Dr. Shayn Peirce Cottler, a biomedical engineer at the University of Virginia. Dr. Peirce Cottler's interest in biomedical engineering began in high school.
My favorite class in school, I had two actually. One was AP Biology and the other was AP Calculus. I also liked physics a lot too. And so biomedical engineering was a field that really brought all of these disciplines together and enabled me to use my skills in math, but also cobble that with my passion for biology and understanding how the human body works.
The researchers in Dr. Peirce Cottler's lab study how blood vessels grow and adapt in different disease settings.
Yeah, the general, theme of my lab is that if we can partner the experimental bench work with the computational modeling work, we can really get the best of both worlds from sort of the scientific standpoint, but also the math and the analytical and the equations and formulas, kind of bring those together into one place, and they can play off of one another and ultimately assist one another, but also in the end advance our ability to form hypotheses and to test hypotheses and to understand how these systems are working and how we can tweak them for a therapeutic benefit.
A main area of research in Dr. Peirce Cottler's lab is the ability of adult stem cells from fat tissue to regenerate blood vessels in instances of disease.
We focus on an adult population of stem cells, so these are coming from liposuction tissue, actually, that is harvested over in the plastic surgery clinic. So this is fat, basically, that would otherwise be discarded. We actually bring it to our lab and we mine it for stem cells. We can grow them in the lab. They're actually pretty easy to grow and they're pretty easy to harvest. We think that they possess some therapeutic potential when it comes to modulating blood vessel growth. So we think that they can be useful in regenerating tissues where blood vessel growth or blood vessel vascularity, vessel density has become compromised. So that's sort of the basis of it. I think what makes it unique is that we're sort of recycling tissue that people are wanting to get rid of, So there's sort of I think the best analogy is sort of composting, right? We throw away our garbage and then reuse it for fertilizer. Same idea with the fat. When we pull these stem cells out of the fat, we actually think that when they were living in the fat, they were associated with the blood vessels in the fat. So it's one way to look at what we're doing is that we're just sort of moving around cells from one vascular bed, aka the fat, into another tissue that needs help with growing blood vessels. So we're injecting them into the eye to help the blood vessels in the retina sort of establish themselves and trend towards a more normal state as opposed to a pathological state, which is what happens in diabetic retinopathy. So that's one example. We've also used these stem cells from fat to try to heal wounds, chronic wounds, or wounds that have trouble healing on their own, to try to accelerate the healing process. The idea, again, being that these cells are very supportive for growing new blood vessels. They secrete things that make blood vessels happy, and they can actually differentiate into a cell type that is a part of all blood vessels. And so we think that they're sort of the building blocks as well as sort of factories that enhance the process of blood vessel growth. So we've seen that it does work in wounds that are otherwise very difficult to heal. These cells seem to accelerate wound healing. We're also investigating their use in peripheral vascular disease. So in the lower limbs, patients that suffer from bad perfusion, basically we're studying the ability of these cells to be injected into the muscle and then differentiate into blood vessel cell types that can then enhance blood flow in the lower limbs. So in our lab right now, all of our work is being done in animal models. That's sort of a starting place when you're developing a new therapy. We hope actually in the next 2 years or so to move to clinical trials. I will say abroad, overseas, this field is rapidly progressing, and there are probably, I think the last time I checked, over 80 clinical trials ongoing with fat-derived stem cells to treat a huge variety of different diseases. I think the farthest along is a trial that's being done in Spain to treat Crohn's fistulas. So these are sort of chronic wounds that patients with Crohn's disease suffer from. And they're in phase 3 clinical trials using fat-derived stem cells to try to accelerate the healing of those particular wounds. But there are really, ongoing trials all over the world right now using this particular cell type to treat many, many different diseases. The area.
There are 3 primary ethical concerns that Dr. Peirce Cottler's lab must consider on a daily basis.
So one area is, we already talked about animal research. And so there's a tremendous amount of ethical obligation we have to those animals to reduce pain and suffering to the extent that we can whenever possible. And so we spend a lot of time trying to figure out how we can do our procedures that in a way that minimizes pain and suffering and distress on those animals, and how we can treat them with the best possible approaches so that they have minimal suffering. The other place that ethics is important is, we talked about stem cells, and the area of stem cells in general I think is fraught with ethical concerns that need to be on the table and examined very closely. So it is definitely a conscious decision on my part to study adult stem cells, actually. I see it as a way to sort of get around the need to use embryonic derived stem cells. That's something that's very important to me, not just as a researcher but as a person, and so that motivation is really fueling my direction in that area. And with stem cells, it's also one of these issues that a lot of work, as I mentioned, is being done overseas using stem cells. And I think it's fair to say that not every lab across the globe has the same level of ethical scrutiny of the types of studies they're designing and how they're treating their patients with stem cells. So there are definitely examples that have been in the news recently where stem cell trials have come under a substantial amount of scrutiny because they are being conducted in patients without the necessary science to underpin them. And so I think that being a researcher here in the United States, we're very lucky and feel very fortunate that the FDA is an integral player in how drugs and therapies get taken into the public domain, but other countries don't have the equivalent standards. And so I think there are corners being cut in the stem cell therapy world. It's dangerous for those patients in those trials in other countries, and it also doesn't set a very good standard for how society views stem cells if they're being used improperly without the proper ethical considerations being maintained. And then the third way is that I mentioned I use human tissue in the lab. And so when we take tissue from the operating room, it's very important that that tissue is de-identified. So we don't know, we have no way to trace back who has donated that fat. And that's an important consideration when you think about the ethics of privacy, patient privacy, and how their information and their tissues are being used, in our case, to promote research, but obviously one day we hope to move this into the clinic, and so it's really important to respect patient privacy and ownership of those patient tissues. Who do they belong to, right? And so there's a lot of ethical issues associated with tissue banking and cell banking, tissue acquisition, HIPAA regulations, and those sorts of things that affect human clinical trials and human studies and the use of human-derived material.
There are strict regulations that Dr. Peirce Cottler's lab must follow when using human tissue for research.
We had to, as researchers, had to sign a waiver that we would not use this tissue in humans, and we would not use it for a financial gain. Like, we wouldn't sell it, right? So we're just literally using the tissue only to study how it can work, right? So as a result, because we're not using it for financial gain, we're not reselling it, we're not repurposing it into other patients, we don't have to get informed consent from patients. So when patients are donating their tissue, they don't even know about it, actually, and it's because that tissue is a tissue that would otherwise be thrown in the trash. That's really the distinction. Yeah, it would be in the biohazard bin, go away and get incinerated. So it's because it's already trash and we don't know who it came from and we're not going to resell it. All those reasons, we can just take it and use it for research purposes in the lab. But if we move to a point where we're injecting it back in humans, or if there's a point at which we want to commercialize any aspect of this, then those rules change dramatically.